Okay, so without further ado, I would like to introduce our morning keynote, Sam Baxter, who represents the Broad Institute of Harvard and MIT. She serves as Associate Director of Genetic and Genomic Data Sharing and is also a genetic counselor. The relationship with her began through the Chan Zuckerberg Initiative, and she's going to touch on prevalence and kind of start to answer the question of how many of us are there. Welcome, Sam.
Hi, everyone. So I get to start off by making you do a little math on a Saturday morning, but I promise you that this is simple math, and that hopefully you will be able to follow along as I go. So I am usually very informal and tell people to ask questions throughout, but I know that that's not necessarily the preference for this, so just save the questions for the end. Hopefully we'll have a little time, and then there'll also be the Q&A. But if anybody has any questions about anything afterwards, find me during the break, during lunch. I love to talk about these things, and always love talking with anybody who has questions about this, so do not fear approaching me and asking anything or having me qualify. So the reason I'm here today is because I've had the opportunity over the last 5 years to work with patient organizations to answer this question, how many of us are there? I will be obviously talking about this specifically to POLR3A and POLR3B today, but many of these questions and these ideas actually apply across a lot of different genetic conditions, and primarily the methods I talk about are for recessive conditions, so I'll dive deeper into that. So I call this project Putting the Pieces Together, because these are all things that I'm talking about that exist in the clinical genetic space. These are all out there. They've been out there for a long period of time, but it's by only bringing the power of data sharing in different ways together that allow us to start thinking about understanding how many people are out there with a rare disease. So, traditionally, the way that we have defined how many people have a particular disease is through something called incidence or prevalence. These are the numbers you often will hear people say. This is how prevalent something is. This is the incidence. Those people who don't have a degree in public health kind of let it wash over them and aren't really sure. But incidence is more traditionally called birth rate, and prevalence is saying at any point in time how many people have a particular diagnosis. The method that I'm going to talk about is something called genetic prevalence, and the reason I always emphasize genetic prevalence is that it's something slightly different. So if we think of these numbers on a spectrum, we're really starting at first with the clinical cases, and there is no patient group or any individual that I've talked to that says this is exactly how many people. If we just go and say, you know, everybody who makes it to the doctor's office and meets exact criteria, that's our number, everybody says we know we're undercounting it. We know we're not getting to everybody, and especially in the rare disease space. So this idea of genetic prevalence has been around for quite a long time. It is actually the foundation of genetic counseling. It's a method called Hardy-Weinberg equation. I will not ask any of you to know this, but basically it's the idea that without external forces, the variants that are floating around in the genetic gene pool stay the same from one generation to the next. And that idea can be leveraged to say, okay, if we know that how often these variants are floating around in the parental generation, the parents, then we can use that math to figure out how often we expect individuals in the next generation, their children, to be born with a particular disease. And so what was missing to kind of do this at full scale were large-scale population databases. And those started coming out in around 2013. And when that happened, people very quickly started to apply this math and started to say, great, we can do this. The problem is that they were overly inclusive of the variants that they wanted to look at. They said, let's include every variant that's ever been identified in somebody with a disease, or any variant that anybody has ever said is associated with the disease, let's put it into the math. And what we realized is that this creates a lot of distrust in the community because it overinflates the numbers very, very quickly. And so when we go back and look at early numbers, quite often they're overinflated. And so our group decided to take a different approach. We are heavily trained in variant curation. This is something that happens upstream of patients getting a test result. There's a whole field of us that are trained in looking at those variants and saying, how confident are we that these cause disease? And this method was missing from the way that people were traditionally looking at genetic prevalence. And so what we did was we came up with a method that, does that large aggregation of give us all those variants that anybody has ever associated with it, but let's curate these variants and feel very confident. And let's give back the patient organizations a range saying, here are the ones we feel we're very confident in, here are the ones that we have questions about, but let's give you a sense of what the bookends are on either side of this, and let's start moving closer and closer down the line to the true genetic prevalence. And the true genetic prevalence we define as the estimated, I know you want to do math too, I get it, the estimated proportion of a population that either has 2 compound heterozygous variants or 1 homozygous variant. The reason I point out this definition, it sounds like a bunch of jargon, is that it's just saying, what we are saying is how many people do we have expected to have inherited 2 disease-causing variants, 1 from mom and 1 from dad. That's not saying whether we know that individual has the disease. That's not saying whether we know that isn't even a possibility that is viable or compatible with life. So we have a very broad umbrella, and we need to think about that when we look at the final numbers and bring that to kind of the front of our brain whenever we're looking at these. So very quickly, I do want to point out, because we've done this with a lot of groups now, this is 1, 2, and 3 is sort of where we find our results with each group. So 1 is the one I never want to give back, but I do sometimes, where the numbers we give them are lower than the numbers that they've already looked at in the traditional incidence and prevalence. And they say, why did we even do this? We knew that, and we know more than what you just told us. But what we were able to figure out there is that population databases and even the databases where we share variants amongst the clinical community are not all-encompassing. We know that there are historically underrepresented populations throughout genetics, be it that they don't have access to genetic testing, we don't know what variants cause their disease, and in population databases, we don't represent every population from around the world. And when you find a disease that is more common in one of those populations, we aren't able to do the math because it looks like their variants aren't even in there. That wasn't something that we had to address directly with this community, but it does happen, and so we need to be aware of that and the impact of representation across the board. 2 is where we think we're undercutting it a little bit with the genetic prevalence. We may not know the full spectrum of variants that cause the disease. There may be a lot of people walking around there that have a clinical diagnosis and we've only found 1 of their 2 variants and we're still missing another variant. And so we just know that the full picture hasn't been put together. And then 3 is where we have the estimate, but it's a little higher than what we think. So we're trying to think about how could we bring this back down, make it a little bit more accurate. But what I hope you understand in what I'm trying to say is that these numbers are estimates and they're beginning points, and that you need to take in the context around you, and that's where the patient organizations come in and their network of researchers. They can come in and continue to refine these numbers, and that's the stage that we're at with your group right now. We have these estimates that I'm going to show you, but I'm going to show you a few places that we already know we need to refine and where we want to continue to kind of bring these bookends in to get closer and closer into that true genetic prevalence. I've already talked about these a little bit, but I like to drive them home for any scientists in the room who are going to end up doing this. These are the 4 things that should always be in your brain. The first is representation, representation, representation. We need to think about who we are looking at, who is missing from the equation, and make sure that we always take that into account. We are working very hard to improve the diversity in population databases and to improve access to all populations across the world for genetic testing, but we are not there yet. And until we are there, we need to talk about this the first time, every time. The next is curation. I have been doing curation for 16 years. The people who wrote the guidelines that we follow are my bosses, so I feel very confident in our curation. But where we draw the lines of which variants to include and not include are still really hard to cover. And so inclusion of a single variant that has a high frequency that doesn't always lead to disease can dramatically impact your results. And so we always need to talk about these edge cases or these variants that we're not totally confident in and make sure that we're thinking about those and whether or not to include them when returning the results. Clinical sensitivity was not as much of something for this particular thing, but again, like I said, if there are certain diseases where not all the variants and the full spectrum of variants has yet to be found, that we need to think about that. And the final is disease spectrum, which has a huge play in what we think about when we were talking to the Yaya Foundation about POLR3A and POLR3B. There are spectrums of the disease itself, and especially across 2 different genes, then you're thinking about different kind of spectrums, and there's some variants that cause a more severe phenotype, some variants that maybe cause something slightly different, and how do we think about parsing this and cutting it and slicing and dicing it? We have a global number because of the gene, but we need to dive in and start kind of building out more refined numbers based off of certain groupings. And that was something that, talking with Ron and Valerie, we knew from the beginning, but we wanted to start with these global numbers to kind of see where the starting point is and continue to dive in. So how do we build this puzzle? Well, we build this puzzle first with 5 core pieces. The first is the sources. Where are we going to get this information? I don't want to just be going to any source out there. I want them to be trusted, vetted, and we know what the biases are of those sources so that we understand that in our model. The next is curating that list. So the sources are going to tell us what variants we're going to include in the math. The curation tells us our confidence in how well we know they cause disease. Once we get to that final list and classification, we debate it heavily for an hour in a meeting, and then we come up with it, and we come up with the ones that we think perhaps are borderline and that we want to dive into more. And then we do the math, which you will all, I promise, follow along in this math, but we do some math, and we get a final estimate. So I'll walk you through each of these steps so that you understand a little bit how we got there. The first, where, like I said, we start at the sources. We want to know which variants are known to cause disease. There are databases that are used in the clinical genetics community that we all work with and are very familiar to us, though I don't think are very familiar to all of you, so I wanted to tell you a little about them. The first is ClinVar. This is an open sharing, open source database where any researcher, clinical lab, even patients can deposit the variants they have or have seen in a patient with a disease and can say what the classification is. Do they think it's disease causing? Do they think it's benign and is just something that we have in our genes through evolution, and give the evidence around it. The next is the Human Gene Mutation Database. This is the opposite of ClinVar. It is paid for, but we pay for it because we need to for our research. And they employ teams of PhDs to scour the literature. And anytime they find a variant, they will put it in the database and list the publication that they found it in. They are very trusting of these papers, and basically anytime a variant was put into a paper, they say it causes disease. That's not how it works. And that's why we employ the curation for both ClinVar and HGMD. But then we also work with the patient organizations, and we ask them, are there any variants that you know about or anything that you want to make sure we include? Finally, we use this database Nomad. So Nomad is the population database that we use. It is currently one of the largest population databases in the world. As of today, right now, it has over 140,000 exomes and genomes from individuals that were enrolled in other studies for large complex diseases like bipolar or heart conditions. And then we are able to use that data for secondary use, not to say we're expecting to find people with 4H in there. We're actually expecting not to find that, but we're looking to see how many carriers there are. So we're trying to look at sort of a normal population. I hate that, that's not the right term for it, but the average population that's out there, and see how many of them are carriers, and see if we can leverage that to then estimate how many people we think would be born with 4H. Well, we know that not every variant has been identified yet. We find more and more variants all the time. That's why ClinVar exists and is always aggregating it. So we went into Nomad and said, We know that variants that cause loss of function, that make the protein not work right, tend to be the variants that cause disease and recessive conditions. So tell us in NOMAD all the variants that we believe may not result in protein function, and give us those variants as well. And we go through and we curate all of these to make sure that we feel confident in what we include. But that's the net that we throw out and cast there. And then we use NOMAD as the source of, okay, now we have our list. How often do we see them? Does that make sense to everybody? Great. So this is a curation page. I don't expect any of you to look at this and understand what it is. It's just to point out that we follow a lot of criteria and we go through a lot of different data points when we cast that net and look at these variants to say, do we think that they are disease causing? And our principle is that we are incredibly conservative. I want to give you the foundation. I want to give you the floor that you can then build up from. I do not want to include a high frequency variant without any caution and give you numbers that then are unachievable in the clinic. This is not what is actually out there. So we employ the ACMG AMP curation. This is the gold standard in the clinical genomics community about how you curate variants. And then we also wrote up a secondary curation protocol that we run where we are looking in NOMAD. NOMAD is a research database. These people weren't sequenced for these diseases. There can be technical errors. There can be things that just weren't looked at because they weren't clinically tested patients. And so we do another sort of more research-based curation to make sure that we feel confident those variants are true and that we're not artificially adding in frequencies that wouldn't really be there. So when we did that, this list that I've been talking about for POLR3A, we had 78 variants that went through this full ACMG criteria. That meant that they had been seen in patients somewhere in the literature, somewhere in a database. And then we had 82 variants that were loss of function, and so they went through this extra curation as well. Many of those were included in the 78 above, but we did this secondary assessment on them. 126 variants made it onto the final list. There were 16 that were called pathogenic, 65 likely path, and 45 uncertain significance, and I'll explain those in a minute. On POLR3B, we had 38 that did the full curation, plus 75 that got additional curation because they were loss of function. 94 made it into the estimate, only 6 made it pathogenic, and 53 likely pathogenic, and 35 uncertain significance. So the numbers, when I give them to you, I'm going to talk about conservative results. We only use the likely path for those conservative results. The variance of uncertain significance meant we weren't quite sure about those. We did return them to Ron and Valerie so that they could understand what those ranges could be. But if I add them in, that is the upper bound. You're not going to get to that number because not all these variants are going to be disease causing. But what can be in there are variants of uncertain significance that maybe are the right ones to go after for research. And so we gave them that list so that they could kind of have a target of who maybe, or what variants, if you need to spend your money wisely, these are the highest frequency VUSs. And so maybe going after those would be a good first start and things like that. So then we move on to calculations. The one thing I do want to point across is that you're going to hear me talk about global, and then you may see in the slides that they're also we call them genomic ancestry groups or subpopulations. Sometimes they're called continental populations. This is genetically determined population, so similar to 23andMe, the way that you give blood and they tell you, where you cluster. Unlike 23andMe, we have to put everybody in one box. That's not how humans work, so it is a bias, but at least gives us a proxy to understand if there's a higher prevalence in certain populations, because that changes the way you then think about sort of the global numbers after that. Because if you only see it in one population, you can't extrapolate that to the world's numbers. You have to kind of keep it a little closer to that population. So it gives us some context. So this is Hardy-Weinberg equation. Anybody who took algebra right now is probably glossing over and hating that they're seeing a binomial equation this early on a Saturday morning. You can skip that. We're going to go down. When you're working in rare disease, we get to take the shortcut. So Q is just adding up how often we see all these variants that we talked about. So adding up the frequency of all those likely path and path variants. So it's just addition. Then we times it by 2 because the Q is how often are these variants floating out there? Well, we get 1 copy from mom and 1 copy from dad, so our carrier frequency, we just times it by 2. And our chance of inheriting 1 from each is squaring it. So I think you all follow me on this math. It's actually quite simple once you get there. A lot of work to get there, but once you get there, it's pretty fast. The crazy Excel sheet I have to use to do this is not so simple, but you don't have to look at that, so that's good. So again, you are going to see the conservative and the relaxed numbers on there, but I'm really going to drive home the conservative very quickly. So here's the part where we get to the numbers. So I will show these in bigger, so I apologize for the small text for those of you like me who have some sight issues. But what I want to take away is that the conservative carrier frequency for POLR3A was 1 in 228, with a conservative prevalence of 1 in 200, just about 210,000. For POLR3B, the conservative carrier frequency was about 1 in 526, and the conservative prevalence was 1 in 1.1 million. But that is just at the surface level. Very quickly, we realized there were particular variants that we needed to dive into to see how much they were contributing to this, and if there was different things that we needed to think about. So for POLR3A, I put this into the bar chart so you can kind of see the ebbs and flows of the different populations. And I don't want to actually harp on any of these populations. This is more of like what I call an ocean graph, where there really isn't any population that's spiking up too high. Even in these subpopulations over here, they're smaller, and so they can see more dramatic. They're a smaller number of individuals, so they can see more dramatic shifts. Overall, there's actually not a huge spike in any given population, but there is one particular variant, the +22 variant, that is known to cause a less severe, more adult onset phenotype. And there is debate, at least in the literature, of whether people who have 2 copies of this can present with some type of disease versus not. And so when we were first returning these results to Ron and Valerie, we were sort of on the fence. Do we include this variant? Do we not include this variant? And I just want to take a moment to show to all of you how drastic these numbers change without you having to know any of this math, to appreciate that going from this to this is a large number of individuals. And so I was a little anxious about, do we include this at all? Do we not? We know some people have this, especially for compound TET. 1 copy is this one +22, and then they have another copy of a different thing that do have a phenotype. So we don't want to remove it altogether. But then again, there is this debate of whether people who only have the +22 2 times have a phenotype at all. So we left it in there to try to show you this is one of the principles of how are we going to handle this? How are we going to refine this math? The next was POLR3B. So we gave these numbers, and as I mentioned, with the POLR3B, the carrier frequency is about 1 in 526. The genetic prevalence is about 1 in 1.1 million. But there was this VUS that wasn't in this conservative number that was bothering me. The evidence was there. It looked like it could really make it to pathogenic, and then sure enough, it has a higher frequency. And so we wanted to highlight that one as well. And that is the MET415 threonine, which again, I'm just going to do this so you can appreciate those jumps up. Those jumps up are potentially people that are out there. So this big of a jump is not something we want to totally ignore. But right now, it's a variant of uncertain significance. So this was another one where, like I said, we give back that list because this is a cool one to go after if you're going to invest your resources and figure out if this is disease-causing or not. If it is, you have a higher prevalence. If it isn't, you could drastically reduce the number of kids or adults who are getting an inconclusive test result. So either way, figuring out the classification of this variant makes a difference to the patient community. So now when we combine these numbers together, we can just kind of appreciate what this is. I'm not going to spend too much time on this because I want to get to the numbers that people do the math. And this is always where I give the big caveat. This is natural, this is normal that people do it. I give you fractions, I tell you 1 in a million, and you just say, what does that mean? How many people? Again, NOMAD is not representative of the entire world's population. So it is hard to do this, but it's natural. So if you're going to do it in my presence so I can give you all the caveats. When we take the frequencies that I gave you, and I even pointed out for POLR3A, after talking to Valerie and Ron, they wanted us to include the +22 in these numbers, that this would result in a conservative estimate of there being 38,000 people in the world with POLR3A related disease. This is not just for H. This is including this broader spectrum, including the possibility that people with the plus 22 and homozygosity have some sort of phenotype. If you take that out and we say, okay, let's take out plus 22, the frequencies, the conservative estimate worldwide would be about 5,500 people and about 231 people in the US. So it does make a huge difference, and we need to dive in and find out what it is. So just know that top number really heavily depends on what happens with this plus 22 variant. The POLR3, this is without the MET415 threonine, and the conservative estimate is around 7,000 worldwide and 300 people in the US. So coming back to this display, sorry, was there anything? Okay. Yes. Coming back to this, how many of us are there? I showed you guys that 1, 2, and 3 position. For this assessment, I think we're at a 3. We're a little bit over the mark where we should be in our initial estimates. And so now it's talking about refining it. But there is a big thing that is about to change. NOMAD, our database that I've been using, which has 140,000 individuals, the thing that is keeping me from sleeping most nights is that I'm also the operations manager for that database, and we are launching in the next month or so the new version, which will have over 800,000 individuals. We know this means that these frequencies are going to change. The very cool thing for the rare disease community is that those variants that before were so rare that they were not going to be able to be picked up at 140,000 individuals and were just added as 0, now actually are going to have a fraction. They're still going to be rare, but when you add those up, we'll get more information. For the populations that are underrepresented, we brought in a database called the UK Biobank. They're all coming out of the UK. We call it the blue wave of Europeans that are added to an already European-filled database, not the best, but with it brought actually quite a few, about 10,000 African samples that came into it. We also have more inclusion from some other studies that brought more admixed Americans from Costa Rica and some, I believe some from South America. So we're actually having a 2.9-fold increase in non-European samples as well, which will give us more power in finding their rare variation as well. So I think that we've got a lot coming in both refining the list, but also improving the data behind it, and that over the next year, we're really going to have improved calculations. So I hope in the interesting trends, what I've been able to show you is that both POLR3A and POLR3B highlight the importance of really drilling in about each of those individual variants, and that one single variant can quite often change the way we think about these numbers, and why the importance of not just doing it, as I said at the beginning, where you just take everything at its word, put it in, get a number, and be happy. You really need to dive in and partner with patient organizations and their researchers to say, how does this feel to you? What are we missing? What do we need to keep including? And so I come back to these puzzle pieces, and when I made this diagram at the beginning of working with all these organizations, I just made something that looked nice, realizing there were missing puzzle pieces, but as I went back, I realized very quickly that what was missing was the inclusion of patient organizations as one of the sources of truth, and that they are now a critical part of this. In the variant list and classification, partnering with the researchers who can provide functional work, and if there's gene novelty, certainly that means there's a lot more to overcome in setting up assays and understanding how often these variants present with disease. In the aggregate frequency and calculations, understanding representation and continuing to improve the population databases behind this so that we can do this for everybody. And then finally, for the prevalence calculations, always talking about these results in the context of a disease spectrum and understanding how that changes the way you think about these numbers. Quickly, the limitations, always have to talk about limitations, is that we can only include the variants that are present in NOMAD. So again, as NOMAD grows, we'll have more ability to include rarer and rarer variants. Right now, we do not include structural variants. These are very large types of changes, but in the coming months, we actually are going to be able to do that, so we're overcoming that quickly. I think I've talked about representation and that not all VUSs are in here, and the main thing also is that a Hardy-Weinberg equation, and thus this result does assume random mating, that any two people in the world could meet up and have children, and while that continues to be better as the world globalizes, we still tend to cluster a little bit in the way that we mate. So there are some biases built in that way as well. And the last thing that I will plug in my last minute is that I don't want everybody to have to come to me for this. I want this to scale, and I want this to scale quickly. And so part of the funding for the Chan Zuckerberg Initiative was I told Tanya, I want to build this tool. I want to build this tool because I promised a mom friend of mine who's also a rare disease parent I was going to build her this tool, and now I have built her this tool. And it's called Genie. It's the Genetic Prevalence Estimator, and it will allow anybody to do the math that I just did. But it will also allow you to share that list with the community of collaborators, and you can do that refinement that I was talking about. You can uncheck the variant. You can add a new variant. You can download the numbers and change the way the math is done, and in real time see these numbers. And then soon next year, we're going to launch it with V4, which will have the improved numbers, and also have an open source database that allows any patient organization or group to publish their list and be able to say, here's our estimates, and here's all of our variants, here's our justification why, and kind of slow down the need to always have a publication to do this, and have an open dialogue on it that people can be able to contact each other and say, I don't think you should include this, or you should have added that. So hopefully you are all as excited, but this is like my third child that I've been working on for a long time, so she doesn't talk back to me nearly as much as the other two, sometimes. So anyway, with that, I want to thank, I get to represent a very large team of people who work on all these databases, my team of curators who curate hundreds of variants every month to do this, and then obviously thank you to the IF Foundation for working with us. They've been absolutely fabulous to work with, and it's been a pleasure, so thank you.